Chapter 21 covers both the lymphatic and the immune system. So we've already discussed the lymphatic system, and now we're going to talk about the immune system. So it was in about 1882 that phagocytes, or phagocytic lymphocytes, were discovered. And in 1908, the Nobel Prize was won for cellular immunity by Metchnikoff, and the Nobel Prize for humoral immunity and immunology was Paul Ehrlich. So we haven't known a whole lot about the immune system and the lymphatic system since in the last 100 years. We've been learning so much about it. And one of the things that I had always talked about and heard about, but never really thought about it until I was taking classes like this, the word oma, if you're in America, means tumor. If you're in Germany, it means grandmother. So when I became a grandmother for the first time, my kids said, "What do you want to be called?" And I said, "Oh, how about Oma?" Because at that time I was living in Germany. And my son goes, "Mom, why would you want to be called tumor?" And so I thought, okay, I'll just go by Grandma Karen, and that'll work out just fine. So if you have a lymph node that has been overtaken by cancer cells, so it's been overwhelmed to the point where it can't kill it as fast as the cancer is growing, then we say you have a lymphoma, or cancer of the lymph node. And we talk about the immune system and we talk about it fighting things, but what is it fighting? It is fighting pathogens. And what is a pathogen? It is a bacteria or a virus or a fungus, a protozoan. So it is an invading organism, or another definition of a pathogen is something that gets in your body and causes you to make antibodies against the antigens on it. Other pathogens that you may have run across are worms and yeast. So I think you pretty much have an idea of what they are. Now, you have specific and non-specific defense. So non-specific is just your skin. It doesn't really care whether it's a worm trying to get in or a virus trying to get in. It's just going to block it all. So it isn't particular about what it fights. And when you get into another line of defense, some of the white blood cells are non-specific. They'll just eat anything that's not supposed to be there. But others are going to have a very specific target. So we'll be talking about both of those. And innate are all the different defenses your body has that you're born with. So you have an innate immune system. But as you age, as you mature, as you're exposed to different pathogens, you're going to develop more and more specific immunity. So that's what this part of the lecture is about. You can get adaptive or acquired immunity after you're exposed to an antigen. So the non-specific, it just starts right in. As soon as it sees something that's not supposed to be there, it starts attacking it. But the specific has to be taught. This is something that you don't want in your body, and you need to start getting rid of it. And it could take, anywhere from 6 to 10 days before it actually starts working, which is why the first time you get the flu, it usually lasts about 2 weeks before you get over it. Slides 49 and 50 are kind of teasers because they say this is what we're going to teach you about. So your first line of defense is your skin and your mucous membranes. So mucous membranes are wet. That would include your nose. Your ears aren't really wet, but they do have a waxy substance in them. Your eyes are wet. Your anus is wet. So you have, and for women, our vagina is wet. So we have mucous membranes and skin that will stop a lot of stuff before it gets inside of our body. Innate, ones you're born with, are your leukocytes, your white blood cells, macrophages, which are monocytes that grow up and get really big and start eating things. And then we have a whole bunch of proteins. Now, this chapter goes through a lot of proteins. If you take a class in immunology, you'll spend an entire semester learning about the roles of the white blood cells, the roles of the antimicrobial proteins. This is just half of one chapter. So I'm going to hit some of the really important ones, and you're welcome to learn all the others, but I'll let you know which ones I expect you to know. And the rest of them you can just learn on your own because they're interesting. But there's a whole lot of them. And then you have natural killer cells. And just the fact of that you have inflammation is going to help you get well. So although we don't like inflammation, we don't like the swelling, the burning, the heat that you get when something is inflamed, that's actually helping you. It's allowing the white blood cells and these proteins to come into the area and help you fix whatever the problem is that you're having, whether it's a specific pathogen or a toxic substance. And then fever. Most people, the minute they get a fever, they run and they take an aspirin, or they run and they take Tylenol to get the fever down. Your body is making a fever because that will help you get better faster. So unless you're just miserable, or unless your fever gets too high. Now, if your fever gets close to 104, 105, yes, you need to bring your fever down. You need to bring that temperature down because that's actually going to hurt you if it gets to 105 or higher. But if you're just running a fever of 99, 100, 101, somewhere around there, leave it alone. It's helping you. But most people we're just taught never to run a fever. And then your third line of defense, we'll spend quite some time talking about that because this is the one that you get after you're exposed to something. So it's not something you're born with. And the other cool thing about adaptive immunity is for most things that it encounters, it remembers it and it makes memory cells so that if it ever encounters that again, you don't catch that again. So that's why we have measles vaccines and mumps vaccines and things like that. So after you're exposed to the vaccine or have the disease itself, then memory cells should remember it, and if you ever encounter it again, it should wipe it out before you get sick the second time. So your skin protects you because mechanically microorganisms and other pathogens can't get through it to get into your body, to get into your bloodstream, to get into your food source. You have keratin, you have hair, you have dead skin cells on the outside of you, you have fingernails and toenails. So these are just, they call them external barriers. It's just hard to get through. The other thing is your outer skin is too dry and it's dead cells, so there's not a lot of nutrients. So there's not a lot for the bacteria to grow on. However, that being said, remember that you have about 10 times as many bacteria growing in and on you as you have cells in your body. So they don't need a whole lot of food. But we don't usually get big growths. Now, of course, if you're a teenager and you start getting acne, then you're going, what are you talking about? We have plenty. But at that time, when you're going through puberty, you start making extra oils and things. And you have testosterone. Even women, girls, will start making testosterone too. So guys make estrogen and testosterone, girls make estrogen and testosterone, but guys make more testosterone than they do estrogen, and girls make more estrogen than they do testosterone. But if you have, once you start making testosterone, a lot of times that'll trigger your acne because of the oil and the on your face. And then you don't really realize it, but you're sweating, you're leaving salts, you're leaving fatty acids, and sebum, all these things that your skin secretes also inhibit bacterial growth. And you do not have to know these other peptides in the skin that kill microbes. Other mucous membranes that I forgot to mention would be the urinary tract. So I did forget that one. And you can go all the way down in your respiratory tract, because not only is your throat moist, your windpipe moist, your bronchial tubes moist, but your lungs are also. So pretty much your entire respiratory system is wet and covered with a lot of mucus, which is nice. It catches a lot of stuff. Your digestive tract, all the way from your mouth, down your throat, through your stomach, and through your intestines, out your anus, the whole way is wet and covered with mucus. So this will capture a lot of things that shouldn't be in there. And then we have an enzyme called lysozymes, and they destroy bacterial cell walls. They can dissolve proteins, all kinds of stuff. So lysozymes are kind of handy enzymes for breaking stuff down and getting rid of it. In the last chapter, we talked about the 5 kinds of white blood cells, leukocyte cells, white cells. And there are some of them that do phagocytosis. That would be the neutrophils and the monocytes. And then you have other types of white blood cells that also kill things. So eosinophils usually are elevated if you've been invaded with worms. The other thing that can cause eosinophils to go up is if you're having an allergic reaction. But generally, if you're having an allergic reaction, it's the basophils that are the ones that are going to move in. And basophils are full of histamine, so they're going to cause swelling. When you think about an allergic reaction, think about basophils, and you can kind of think they're responsible for a lot of the misery that you go through. And monocytes, when they're little, they look like lymphocytes, and they can be confused. These two can be confused. But as soon as the monocyte is exposed to something that is that needs to be eaten, it becomes huge. It just becomes huge. And then there's no mistaking it for a lymphocyte. And lymphocytes are kind of cool. We're going to spend quite a bit of time at the end of this talk talking about B lymphocytes and T lymphocytes, because they're the ones that make the antibodies. So you've been hearing all kind of stuff about we're having COVID vaccines, and we're checking to see if people are making antibodies. We're checking to see how long the antibodies last, and checking to see, do the antibodies protect us if we're exposed to COVID a second time. So we're going to spend a little time talking about that, because I think everybody should be interested in that at this point in time. So starting out with the first of the 5 types of leukocytes, the neutrophils are phagocytic. They run around eating things. They do phagocytosis. And the biggest thing that they do that's kind of cool is they go into an area where there's a lot of bacteria, and they will start eating them. So you'll actually see the bacteria inside the neutrophils as they're eating them. But then they also produce a cloud of bacterial chemicals, bactericidal chemicals, that'll kill the bacteria. The problem is, if you put all of this stuff out there that will eat up the bacteria, it also kills you. So the neutrophils last 1 to 3 days, and then they're dead because they've eaten and destroyed, and then they just go out in a big burst of chemicals and enzymes that kill in a big area around them, but they die also. One of the things that we're going to find out about the 5 kinds of white blood cells is if one of them is stimulated and starts attacking a foreign invader, it usually sends out chemicals that summons other white blood cells. So in the case of eosinophils, if you have tapeworms, roundworms, other kinds of parasites, but usually we usually think of worms. If eosinophils go up, they go, let's go check for worms. And they make chemicals, superoxide, hydrogen peroxide, and other toxic proteins that can kill these larger things. Tapeworms and roundworms are visible to the naked eye, whereas bacteria are not. And they also call in the basophils and activate them, and the mast cells. So basophils and mast cells are both famous for secreting histamine, and histamine is what causes fluid to build up in an area. So if you have swelling, this is usually caused because the basophils and the mast cells are releasing histamine. And to get the swelling down, of course, you would take an antihistamine, such as Benadryl. The other thing that eosinophils can do is, we don't know what an antigen-antibody complex is yet, but when we get to that part, eosinophils have the ability to eat those. So that's good. You just cover something that you don't want with antibodies, the eosinophil comes and it finds it, and it eats the whole thing. It eats the invading thing that's got antigens as well as the antibodies that are covering it. Other things that the basophils do besides make histamine, which causes fluid to come into the area and causes vasodilation. So the blood vessels open up, so you get more blood flow in the area. So it's easier for the other white blood cells to come into the area because you've got more blood flow, you've got larger blood vessels, and you've got fluid in the area. The other thing it makes is heparin, and heparin inhibits clot formation. So if you've got an area where you have damage going on and you're trying to make clots, if the basophil comes into the area and produces heparin, it's going to slow down the clotting. And mast cells also secrete a lot of these leukotrienes and other things like that. We take the lymphocytes and we break it down into 3 categories. The B and the T are going to be the ones that are responsible for antibody formation, but the third type is natural killer cells, and so they just call it NKs for short. Natural killer cells, they are part of your innate immunity. You don't have to wait and be taught that you don't like a certain thing. As soon as a natural killer cell sees something that is foreign to your body, it just kills it right away. So you don't have to wait for days to make antibodies and go through this whole process. So the B and T cells are considered to be adaptive immunity, and the natural killer cells are innate. You're born with it, and they're ready to kill right from the get-go. And they're great because they also kill cancer cells. They kill your cells that have been invaded so that they don't allow the cancer to spread, or if the cells have been invaded by viruses, it doesn't allow the viruses to escape the cell. It kills the cell which was making the viruses. So natural killer cells are really handy things to have in our body. Monocytes have the ability to leave the blood vessels and go out into the connective tissues and eat things there. And remember, monocytes turn into very, very large macrophages, which are able to eat considerable amount of invading pathogens. So some of them move around. They're all over your body. Some of them stay put. So they just stay in one place, and then anything that happens to come in that area, then they eat them. So some of the more famous or well-known of the macrophages are microglia. That protects your central nervous system. So that's your brain, your nerves, things like that. So microglia are good. So we don't want stuff growing in our brain, obviously. Alveolar macrophages are in the lungs. Most people call alveolar macrophages dust cells because they kind of look like tumbleweeds as they're moving around inside of your alveoli. So your lungs are like a bunch of grapes. If you look at it from the outside, it looks like grapes, but if you get on the inside, they're hollow, and that's to allow air to come in and be absorbed through the cells. But you also, anytime you're breathing in spores, you're breathing in pollution from the car in front of you or the bus in front of you. So you're getting all kinds of yuck that are pulled into your lungs with every breath. So these macrophages are running around eating the stuff that you breathe in. And then you have some macrophages in your liver too. So we've talked about the 5 kinds of white blood cells, and I promise you that we'll come back and talk about B and T lymphocytes when we talk about antibodies. But before we get to that, we're talking about short-term innate immunity. So this is something you're born with. You don't have to be trained and learn how to do this. 2 of these are interferons and complement. Now, I'm going to tell you a little story about interferons because it's one of those mysteries where you're like, man, I wish I knew the end of that story. But once upon a time, about 40 years ago, I was hired to run a grant that this doctor had written. And at this time, they had just gotten really excited about interferons because what an interferon is when a cell is invaded by viruses, it's too late for that cell. It's already been invaded, its machinery has been taken over, and it's cranking out viruses. But what it can do, as the cell is dying, it can make interferons, and it sends them over to all the nearby cells, and as soon as they come to the nearby cells, it tells them that there's a viral infection that's coming. And interferons are specific. So one interferon won't protect against 2 different viruses. So like if you have interferons against chickenpox, it's not going to help you against measles. So those are different viruses. But if it is interferon against chickenpox, for example, it's going to tell all the cells nearby that chickenpox is coming. And so they're ready, and they will not allow the virus into their cell, so then it can't take over the machinery inside the cell and make new viral particles. So interferon is an amazing thing. So the grant that I was working on was, first of all, we needed a quick test to find out which kind of flu a person had. And so you've probably been hearing a lot about the different kinds of flu, and they have a number, an H number and an N number. So like H1N1 is it's called swine flu, and it's scary. It's the one that caused the millions and millions of people to die in 1918 in that flu epidemic. And so everybody's comparing this coronavirus with the flu of 1918, and there's no comparison between the two. But anyway, that's a whole nother story. So what my job was to get every single flu that had been out, and in the population, because anytime the flu goes through, there's always laboratories that will save samples of the flu. And so I found a laboratory up at the University of Cincinnati that had samples of the flu all the way back to 1941. So that was way before I was born. And I got a sample of each one of those, and I raised antibodies against each strain of flu from 1941 until the grant that I was working on, which was around 1982, somewhere around there. I was working on the grant. So once I had antibodies, then what I could do is I could take these little plastic beads and I could coat them with antibody. And then I would get a sample from a patient who was sick or in this case, I had samples from patients from 1941 even. And I would put a little bit, well, first of all, I would sonicate the sputum sample and get the viruses out from the mucus that was in there. And then I would put my little bead with the antibodies in there, and the antibodies would stick to the virus if it was the correct virus. So it was a screening process to see if my antibodies could detect which of the different flus that was there. So once I had done that, then I had I was using radioactivity at the time, but you could also use a color protein. And if the antibody matched up with the flu that I happened to put in there, then I'd have a positive test, and I would say, well, that's the WSN virus, or that's the H1N1 virus, or, whichever type of flu that I had. So the idea was, if we had a quick test, so if somebody comes in, they're coughing, they've got the flu, they've got a high fever, they're miserable, you just have them cough some stuff up into a test tube, and you drop one of these little beads down in there, and you would see which strain of flu that they had, and then you would give them a shot of interferon specific for their flu. And it should have worked and should have wiped it out. But the thing is, the method has been adopted for a lot of stuff, but there's been no interferon shots developed for the flu. So I'm a little bit surprised about that. But like I said, I did my part of the grant. I made the rapid diagnostic test and showed that it was specific for each of the different kinds of flu. And then I passed it on to someone else to take over the interferon part, and I never heard about the rest of it. So we still have those quick, rapid tests to this day, but instead of using little beads like I did, they use little plastic sticks. So I'm sure that especially the ladies out there who are listening know about peeing on a stick and it turning colors, and you go, oh, I'm pregnant. So we have a lot of rapid tests now that on little plastic sticks instead of using the beads like I did. So not only does the interferon protect nearby cells to keep them from being invaded, it also activates the natural killer cells and macrophages so they can come into the area and eat the viruses that came out of the infected cell. So the object is to destroy infected cells before they can release the viruses, and to send the interferon out to keep any other cells in the area from taking the virus in and starting to make new viruses. So interferon is very powerful. The complement system is also really interesting. It's a group of different proteins, and so they have like C1, C2, C1A, and I'll not ask you to memorize all of the different complements, but realize that there is a complement system out there. It's made by the liver. It runs around in your blood in an inactive form. So it's just ready for a fight. It's like it's got its guns ready, but it's not doing anything. It's just circulating, waiting for a fight to happen. As soon as it sees a pathogen, it is activated, and then you start a complement cascade. So complement does its thing by 4 different pathways. So one of them is the inflammation that brings other things into the area to help kill. It helps with immune clearance, and we'll talk about that in a minute. It helps in phagocytosis, and it actually helps in poking holes inside of cells, which causes their contents to leak out. So cyto means cell, and lysis means to break open. So you literally break the cell open using complement. And then we have 3 different ways that really we're not going to get into a whole lot of that. We'll talk a little bit about the classical, and the alternative pathway meets up with the classical. So we'll talk about that. Kind of an overview of the classical pathway. If an antibody has attached to an antigen on some pathogen, so some virus, some bacteria has attacked you, and you make antibodies against it, then as the antibody combines with the antigen, then the antibody shape changes, and on the antibody is a place where there is a complement binding site. And if you bind C1, then this sets off that cascade that I was talking about, and you end up with a chain of complement proteins attaching to this antibody. So here's a picture, which is kind of interesting because this is not the same one that's in your book, but it's similar. So this is called the heavy chain of an antibody. Right here, there's one of those, and then there's one of these. So there's 2 of them. And then there's the light chain. So in all, there's 4 of these proteins that are stuck together like this. And all antibodies pretty much have the same area right here. This is pretty much similar from one to the other. The variable part is this right here. And this is going to be specific for measles or mumps or chickenpox or coronavirus or whatever you come into contact with and you can grow antibodies against. Now these little bumps right here, which they forgot to label, are the complement sites. So it's said that once an antigen has bound onto an antibody, so you have this antigen and this antibody, it's going to change the shape of this, and now complement is going to come in here and bind to these complement sites. So the classical complement pathway requires an antibody to do its thing, but the alternative pathway is nonspecific, so you don't have to have that antibody. But so you're going to go ahead and have that cascade, and you're going to start with C3 instead of C1, and you're going to go on down, and you can target human tumor cells. So you can attack your own cancers as well as viruses and bacteria and yeast. And then there's a lectin pathway, and this is where you're using plasma proteins. So, the plasma that's running around your blood, you have proteins in there that can bind to carbohydrates. And these can bind to certain sugars on the outside of bacteria. So we don't really think about bacteria as being candy-coated, but they do have some specific sugars on the outside. So if you can get complement to bind to those sugars, then you're going to set off another one of those cascades where we're going to start killing things. So let's talk about how do we do that. As part of that cascade, the C1 or starting with the C3, you're going to get a form of C3 that will stimulate mast cells and basophils and cause them to secrete histamine and other inflammatory chemicals. So complement kind of eggs the mast cells on and the basophils on to make and release histamine. It also, if you have complement in the area and that cascade is occurring, you're going to attract neutrophils and macrophages to the area. And anytime you have inflammation, it speeds up pathogen destruction. So while we view inflammation as, oh my goodness, I have all this inflammation and swelling, it can often be a good thing. It's what you actually want to have happen. So that's what's going on with the inflammation. You're basically you're irritating mast cells and basophils into producing histamine, releasing that. Now, immune clearance. So you've got this antigen and this antibody, and it flexes and allows the complement to bind to it, and you get the cascade going, more complement binding, and it will go through attached to red blood cells until it reaches the liver and the spleen. And there, the liver and the spleen know how to pull the antigen and the antibody off without hurting the red blood cells. So the red blood cell didn't even know that it was a conduit, that it was carrying this around. So this is kind of a cool thing that complement knows how to do. It says, come on, I'm going to take this antigen. Now the antigen, we need to probably stop just for a second, talk a little bit more about an antigen. I talked about it on the last tape, but I'm not sure that you were paying attention. So I'm going to bring it up again. So I talked about the blind people that were feeling an elephant, and every one of them reported that an elephant was completely different than the other one. The one holding the trunk said the elephants are shaped like a snake. The one that grabbed hold of the legs said, no, they're shaped like a tree. They have these great big trunks like a tree. And the one that grabbed hold of the ears said, no, it's a flat, floppy thing. And the one that got a hold of the tail said, no, it's a hairy, bristly thing on a stick. So every little bit of an elephant is a little bit different. And every little bit of a virus is different. Every little bit of a bacteria is different. So each of those different things, the trunk of the elephant, the tail of the elephant, could be an antigen. So an antigen is just anything that can cause the body to make antibodies against it. So it doesn't matter if it's a part of a worm, if it's a part of a virus, part of a bacteria, a fungus, it doesn't matter. As long as it's large enough that the body can see it, recognize it as a foreign invader, and make antibodies against it. So here I have the antigen, and the antigen may be still part of the worm. It could still be part of the virus. It could still be part of the bacteria. And there's the antibody that's joined to it, and then the complement joining to that. And so you're just getting this big mass. It's stuck on a red blood cell, and it goes to the liver and the spleen, where it just strips it off and kills it, gets rid of it. So this is one of the main things that you do, because we know everybody says, oh, we've got to raise antibodies against COVID. Well, what do you do after you've raised antibodies against COVID? You're going to have to send those antibodies in after the viruses. They're going to have to stick onto the virus. The virus is going to have to go somewhere where it can be eaten. It can be destroyed. So one of the ways that you do it is if complement binds to it, then it goes down into the liver and the spleen, and they take care of it for you. So immune clearance. What do you do after the antibody sticks on? What do you do after the complement sticks on? Sometimes it seems when you have a teacher talking about all this stuff, it's just like big words, blah, 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 blah. So on this one, if you look, you've got words like opsonization and, C3b. And you're like, what the heck? So your book tried to be funny, and I thought it was kind of interesting. It says one of the things that complement does is it coats the microbial cells. So it literally attaches to the microbes so that the neutrophils, macrophages, the other things that will eat things, that will phagocytize things, will find it attractive and appealing. So it said it's like it's buttering up to make the foreign cell more appetizing. So the neutrophils and the macrophages don't just go out and grab a bacteria. It needs to have complement coating it. So opsonization is coating of a pathogen by complement. And then the phagocytosis happens. So anyway, I thought that was funny. And this is my favorite thing that complement does. You're going to have a complement protein, and it's going to stick through the phospholipid bilayer, and it's going to recruit another one, and it's going to stick through the phospholipid bilayer. So here you have the different complements, and they make a ring. Unfortunately, it's big enough, it holds the membrane open, so the guts spill out. So they literally poke holes in the membrane of whatever the invading organism is, whether it's a worm or a bacteria or yeast. So we've spent some time talking about interferons, which are proteins that come out and warn other cells that a virus is in the vicinity and to beware of it. And interferon is specific for one virus only, not a whole bunch of different viruses. Complement, on the other hand, is happy to kill all sorts of stuff. So it coats all sorts of things. And if you can make an antibody against it, complement will jump right in there and help you with immune clearance. So, but like I said, my favorite is where they poke holes in. So natural killer cells kind of do something similar. They recognize foreign cells and they kill them. So they can kill bacteria, but they also, if somebody gives you a kidney, donates a kidney or something like that, you can reject it. And the way you reject it is your natural killer cells see those transplanted cells from somebody's kidney or someone's heart, and it goes, no, that's not part of you. And so then it goes and it starts blowing the cells up. So you're going to have to kind of squelch the natural killer cells. Otherwise, unless you get a transplant from an identical twin, there's going to be some of the whatever it is that's transplanted into you that's different. And you don't want the natural killer cells to kill the kidney you just got transplanted or the heart. So if a cell gets infested with viruses, infected, and they're dividing inside there, the natural killer cell can come and kill the whole cell, including the viruses and cancer cells. So that's one of the problems you have. If you have a transplant, then you've got to kind of squelch the natural killer cells. But if you develop cancer, you need those natural killer cells. So I would not want to be the doctor who has to decide how much to knock down the natural killer cells. Once the natural killer cell recognizes something as a foreign invader or one of your own cells that isn't working correctly because it's turned to cancer or it's got infested or infected with some bacteria or something, virus, then it's going to release proteins called perforins, and they're also going to make a ring and make a hole in the plasma membrane. So they kind of, I guess, I don't know whether they learned it from the complement or the complement learned it from them, but they poke holes. And then not only that, but natural killer cells actually have enzymes that they can put through the pore and cause the cell to kill itself. So the word for a cell killing itself is apoptosis, or programmed cell death. So here is your cartoon, and it shows the natural killer cell attaching to an enemy cell for whatever reason, poking holes, sending out porphyrin to poke holes, and then sending out its enzymes inside there, and the enemy cell dies. It blebs up. These are called blebs. So here it's nice and smooth, and here it's all funky looking, and then it just pops. Now, if there's a macrophage in the area and it sees a cell that's dying, then it'll say, oh, that's nice, and it'll go ahead and eat it and get rid of it the rest of the way. I've never quite figured out why, if a person is laying in the hospital bed, that they say, oh, the patient is febrile. It's like, why don't you just say they're running a fever? But anyway, so you'll find this written in their chart. And why do you have a fever? Well, there's all sorts of things that can cause fevers. We know for sure that flu causes fevers, and we know for sure that COVID virus does. But there are other infections that you can get that will cause you to have a fever. There are brain tumors. There's an area in your brain that regulates your body's temperature. It's the hypothalamus. And if you get tumors in that region, then it's going to, your body doesn't know how to set its temperature anymore. That's pretty rare. And oddly enough, cocaine acts on the hypothalamus. And so a lot of my students have been, they are in the emergency vehicles. They're first responders. And a lot of times they'll find people dead from a cocaine overdose, and their temperature is like 107 or 108. So if the cocaine didn't kill you, the high temperature certainly would. Anyway, I didn't realize that that was one of the things that cocaine could do to you, was to raise your temperature so high. So the Native Americans, the Indians, knew that fever would oftentimes cure whatever was invading your body. So they'd have sweat tents that they would make, and they would heat up rocks and then pour water on it. And the person that was sick would be in there, and the steam and the heat would raise their body temperature like you would as if you were getting a fever. And a lot of times that would cause the person to get well better. So fever in moderation does more good than harm. It increases interferon activity. It increases your metabolic rate. It increases your rate of tissue repair. So what a lot of people don't realize is when you're invaded with a foreign invader, it's going to be eating your cells. It's going to be, the viruses we know take over cells, and the cells die making copies. So you're going to have to replace the cells that died. So worms, they're busy clomping onto us, attaching, eating our cells, eating our nutrients. So you're going to have to repair. You're going to have tissue repair after something has invaded you. And fever inhibits the reproduction of bacteria and viruses. So bacteria and viruses have, well, especially bacteria, have a temperature that they like to divide. And if you go over that temperature, then they don't want to divide. If you go under that temperature, they don't want to divide either, which is one of the reasons we put our food in the refrigerator, so that it stays cold enough that the bacteria don't divide. Or we cook it and kill the bacteria that way. There's a lot of material on this slide, but we're talking about fever. And you need to know that if you're running a fever and you want to break the fever, you're going to have to block prostaglandins. And specifically, this prostaglandin is causing you to have a fever. So that's how aspirin and ibuprofen work, by inhibiting this. Now, what causes you to have a fever, what causes you to be febrile, is you can have exogenous pyrogens, meaning outside of you. So that would be like a bacteria or something that causes you to run a fever. But you also have endogenous, which would be the neutrophils and the macrophages. So to help them, they're going to release chemicals that cause you to run a fever. So you can do it to yourself, or you can have some invading organism, like a bacteria, that causes you to run a fever. And like I said, the hypothalamus is what sets your body temperature. So some people, if you take their temperature, they're 98.6. So they've said, well, the average is 98.6. But some of us are running around, and my normal body temperature about 96 instead of 98.6. So if I were to actually go up to 98.6, in my body, I'd be running a fever because I don't usually do that. But anyway, my hypothalamus just decided that 96 is where I need to be, and so there I am. Now, you all have had a fever before, I'm sure, but you may not have analyzed what's going on. So on the onset, so you're just developing the fever and your fever's going up, you'll start shaking. So you have the chills and you just feel really punk. You just feel really bad. So what you're doing is you are causing the muscles, when you're shivering, you're causing the muscles to contract, and anytime your muscles contract, you're generating heat. You also redirect blood flow, so certain areas get hotter than other areas. I don't care if you memorize this or not, you're welcome to do so, but you have a normal body temperature, something invades your body, your temperature goes up, you got the chills, you got the shakes, and then finally it levels off and stays at a certain temperature for a while. And then once you finally killed off whatever it is that caused your temperature to go up, then you drop back down to normal. So, I mean, that's kind of a duh. If you walked into anybody's house in the past and looked in their medicine cabinet, they'd have a bottle of aspirin. Everybody took aspirin. It was the go-to thing. It got rid of headaches. It knocked your fever down. It was wonderful. But in a few kids, usually prepubescent kids, if they got sick with, especially with either the flu or chickenpox, and you gave them aspirin, in just a handful of them, didn't happen to everybody, and you really couldn't predict who was going to do it, but you give your kid aspirin to make him feel better, and you end up killing him. So aspirin can cause something called Reye syndrome, R-E-Y-E, Reye syndrome, like the rays of the sun. And it causes the nerves, the nerve cells, the neurons in your brain to swell up. It causes fatty infiltration of your liver. But the biggest thing, the biggest problem you have is brain swelling, because your brain is encased in a hard skull. So you can't swell. You can only just swell so far, and then you end up crushing the brain, because it can't go any further because it's run into basically a brick wall. It's run into the skull. So there's anywhere you can go. As the brain is swelling, then the child is going to be vomiting. They're going to be disoriented. They don't know where they are, what's happening. They may start having seizures. They may lapse into a coma, depending on how much their brain swells. But about 30% of the kids who get Reye syndrome are going to die because of this brain swelling. And a lot of the ones who don't die end up, it destroys their mental capabilities. So they're going to have mental retardation. They're not going to be able to function or learn, probably not go to college, things like that. So the take-home message from this slide: never give aspirin to children with the flu or the chickenpox or pretty much any high fever. Nowadays, we have Tylenol, and it does not trigger Reye syndrome. So it didn't happen to a lot of children, but it happened to enough children to where they just say, just don't even bother giving it to children, since we have Tylenol that they can take instead. I'm sure you guys all know the 4 things that indicate that you have inflammation. One of them is the area turns red. It swells up because you're releasing histamine in the area. Because you have the swelling and the blood that comes into the area, you're going to have heat. And then you got that pesky prostaglandin that's going to be causing pain. And most of us feel like pain is not a good thing to have. But if you didn't feel pain, you wouldn't know that something was going on. So that's one of the reasons a lot of people die of pancreatic cancer, because most people think that cancer is painful. But unless it's in your bones or something, cancer actually doesn't usually hurt. So it would be nice if cancer did hurt, because then you would say, oh, I'm hurting. Let me go check and see what that is. And they could catch the cancer early on. But a lot of cancers have already spread throughout the body before you even realize that you have it. So pain is actually a good thing because it tells you something's wrong. If you see the suffix -itis, it means that you have an inflammation going on. So you have arthritis, was inflammation of the joints, pancreatitis, inflammation of your pancreas, dermatitis, you've got something like poison ivy. So itis just means an inflammation of tonsillitis, inflammation of your tonsils. So what is causing the inflammation? Well, there's a group of chemicals called cytokines. And I wanted to bring this up because there are people who get COVID. It's like the common cold. They have it a few days, they feel a little punk, and they kind of, drink orange juice, take vitamin C and D and zinc, and then in a few days they're fine and they can go back to work as soon as they're cleared to be able to go back to work. And other people, they have what we call a cytokine storm, where the body, instead of just turning on enough cytokines to kill off the COVID virus, just goes nuts and starts killing off a lot of cells. In fact, they kill off so many cells that the people end up dying. So the cytokines running amok is kind of a curiosity. It doesn't happen in everybody, but if it happens, then the person is in mortal danger from the cytokine storm. So as you're looking through your book and reading about it, keep in mind that this is kind of an unusual thing that we're seeing, where people getting COVID it triggers a cytokine storm, but not in everybody, in just a handful of people. So cytokines mostly are made by white blood cells, and whatever cell is bathed in the cytokines that are made by the leukocyte, it changes the way the cell works. So it kind of takes over and says, okay, you've been doing it this way, but I want you to do it this way now. What the cells do and what the cytokines do usually is short range. So it's nearby cells that are affected. And so we talked about interferon. As interferon is dying, as the cell is dying, it sends out interferon and it coats or goes to the cells nearby, and they're instructed by the interferon not to allow the virus in. So short range, it works in that area. So that's why, again, why are these cytokine storms happening throughout the whole body? Because that's not the way they work. They just work in this area to clear out the infection in this area, which is the way it happens in most people. but some people they're like hypersensitive. The only thing I could think of, I was trying to think of an analogy, is if I get stung by a bee, then I'm really irritated because it hurts, and I have to pull the stinger out, and then I have to limp for a little while because it takes a few days before it heals up where the thing stung me. So that's what happens when I step on a bee. But other people, if they stepped on the bee, they would go into anaphylactic shock and die. So there are a lot of people that are allergic to wasps and bees, and it could be a death sentence if they got stung. So I'm wondering if they could figure out why some people are susceptible to cytokine storms and most everyone else is not. So anyway, interleukins, interferon, tumor necrosis factor. We've not talked about tumor necrosis factor. This is a chemical that's released that kills cancer cells. Necrosis means die. Necropsy is the study of dead things. So tumor necrosis factor, kill tumors. Chemotactic factors, these are things that summon other cells into the area. So there's a whole lot of them. Like I said at the beginning, there are a bunch of different chemicals that they're talking about in this chapter, and I would like you to know what interferon is. I'd like you to know a little bit about complement. Obviously, I don't want you to know all 30 complements, and I don't want you to memorize the classical and the alternative pathway. I have a few pictures to show you. Here is inflammation. Obviously, this is not good. This is rather widespread inflammation. Here are natural killer cells attacking a cancer cell. So here's the cancer cell, and there's your natural killer cells poking holes in it and putting digestive enzymes inside that'll cause it to bleb up and die. Here's a picture of red blood cells and two white blood cells. This one's a lymphocyte, and this is a macrophage. Well, it's not a macrophage yet, it's a monocyte. So before it's angry, before it's activated, you can see how they could easily be confused. This one is almost the size of a red blood cell, and this was only slightly bigger. So it's nice in this particular blood sample to have two of them side by side so you can see how incredibly close they are. But once this monocyte has been activated and becomes a macrophage, it will be almost this large. So it really gets big when it's activated. Here's another picture. This is a lymphocyte right there. The nucleus is almost as big as the cell itself. And then these three are neutrophils, which most of us call polymorphonuclear leukocytes, or PMNs, because the nucleus, poly means many, morpho means shape. So these are many-shaped nucleus, polymorphonuclear leukocyte. Leukocyte just means white blood cell. On the last conversation that we had, I showed you diapedesis. So some of the white blood cells will actually crawl down the blood vessel. They're being attracted to some chemicals that are being released, and they're being summoned. When they get into the area, because you have vasodilation from the histamine, and you have fluid in the area, and you have extra blood coming into the area because you have the swelling, you are able, the white blood cells are actually able to crawl through the skin cells and get down into the tissue where the bacteria have colonized, or the splinter that you stuck in you has brought in different germs, toxins, whatever, dirt, and pushed it in through your skin. So something's got to get in there and clean up anything that starts trying to grow on the end of that splinter or thorn, whatever. Another thing besides the prostaglandins that causes pain are bradykinins. So that's another one of the chemicals that'll cause you a lot of pain. And we know that the name for swelling is edema, and the redness that occurs is because you have more blood flow. Hyper means too much, and so this is too much blood flow into the area. Here's a cartoon drawing showing where somebody stuck a splinter in through their skin. So you bypass this barrier that kept all the bacteria out, and you push these bacteria in on the tip of the splinter. And so now what you're going to have is, here's your blood vessel that's running nearby, and the white blood cells are literally crawling out. They're pushing their way through the capillary walls, and they're crawling over, and they're going to start eating these bacteria. So when you damage the tissue, you send out chemicals that are calling these mast cells to release histamine, to release anti-clotting agents, and then the polymorphonuclear leukocytes, or the neutrophils, will crawl over diapedesis and crawl through and go do phagocytosis. So there's your chemotaxis. This is being summoned by chemicals. So neutrophils are on the job within the hour. They're very quick to come to an area. As soon as the chemicals are released, they come running. Quite fast, actually, for a white blood cell. So other things that neutrophils do besides eat the bacteria and do that respiratory burst, which is where they just spew out digestive enzymes, which kill the bacteria that are in the area, but it also kills the neutrophil itself. Before it dies, it releases cytokines. Other macrophages are going to come into the area because they're recruited. Other neutrophils will come into the area because they're also attracted. And these macrophages and T lymphocytes are going to come into the area, and they're going to cause you to make more white blood cells. So if you go to the doctor and they take a blood sample and they say, oh, you have elevated white blood cells, you know somewhere in your body you have a war going on. So they need to find out where the war is and what is it fighting. Is it a bacterial infection? Is it a viral infection? Or is it a cancer that they're fighting? So usually if you have neutrophils that are elevated, it's because you have bacteria. Usually, if you have eosinophils that are elevated, it's because you're either having an allergic reaction or you've been invaded by worms. So the neutrophils are on the scene in about an hour, and then within about 8 hours, you're going to send in the monocytes, which have turned into macrophages, and they're going to be on the cleanup crew. So they're going to continue eating the bacteria, but they're also going to clean up any damaged host cells and any dead or dying neutrophils. So waste not, want not. I'm always trying to teach you new words so that if you're out at a party, you can be the life of the party talking about these things. So pus is what is formed when you have a bunch of dead neutrophils and the bacteria and the cellular debris, because the cells nearby are dying because of the neutrophils, and then the fluid in the area. So that whole mess of bacteria and dead white blood cells is called pus. And an area that's full of pus is called an abscess. So you can have an abscess tooth, you can have abscesses on your skin, you can have abscesses in your intestines or in your mouth. So it's just a great big bag full of pus. And sometimes you can get enough pressure on an abscess that it can explode and spew pus everywhere. And there is a lady who has a really popular YouTube following, and they nickname her Dr. Pimple Popper. In each of her videos, she shows draining abscesses or exploding abscesses. So if you want to know more about abscesses, apparently this is a highly enjoyable, highly informative series of YouTubes. Dr. Pimple Popper. I'll stop here and let you guys go watch the Dr. Pimple Popper show, and I'll be back with part 3 of chapter 21, where we talk about specific immunity, how the B and T cells protect us against viruses.